Okay, here we are, chapter 5. It's about viruses. And hey, we're in the middle of a global pandemic caused by a virus. So this is a great thing to learn about. So I just put this quote up just because I think this is so fascinating, right? The idea here is that a flea, right, which we consider a little parasite of ourselves, right, has some smaller flea that preys on it, right, some other pathogen, and even those have smaller fleas on them, right? So this idea that there just keep being these smaller and smaller forms that seem to be able to prey on hosts, right? So we were even just recently talking about bacteria. One of the things we'll see in today's lecture is actually viruses that infect the bacteria that are inside of us. So yeah, it's kind of this theme in nature. So one of the strange things that happens with viruses is there's like this big debate. I don't know that people are still duking this out, right? But like you get into different camps of scientists who like to argue about whether viruses are living or not. And so there's a group that says, you know what? Viruses aren't living. They don't exhibit any of the properties of life. So if you remember in Gen Bio, you'd go through things like, life has to be able to metabolize, grow, reproduce, respond to its environment, maintain homeostasis. All of those things are really done by its host cell. So one group says, you know what? These are not living. And then the other group says, okay, yeah, but this is kind of weird, right? Viruses are actually like directing cells to produce the proteins they want, right? So in some ways, they're like almost like above life, kind of like running, I don't know, like a little marionette. That's what I was doing there. Right? So they can direct cells to produce what they want. Clearly, they're more than just like these inert, lifeless molecules, right? So one camp ends up saying, hey, they're infectious molecules, and the other says, they're more than that, right? They're not just lifeless, they're possibly even controlling life on Earth. So, where your book kind of falls out on this is they say, okay, let's go with this as our working definition here, right? They're infectious particles that are obligate intracellular parasites. Now, if you break that down, it's not quite as bad as it sounds, right? Obligate. They must live within another organism, right? They have to do this, right? You know, a parasite already. Whoop, where's my little pointer? A parasite is something that lives in or on a host. So they're obligated. They must live in or on a host, okay, as a parasite. And then this term intracellular, remember, intra is within, right? So they're parasites that must live within cells. Okay. So we know viruses that can infect animals. There are viruses that infect plants, fungus, protists, even bacteria, right? And they can actually carry genes from one host to another. And they have likely influenced the evolution of all life by this kind of transfer of DNA around between different organisms. So if you spend too much time thinking about this, you're like, wait, I'm not even human. I'm just like a carrier for bacteria, right? We were talking about that last week, right? It's part of this mutualism, and there's 10 times as many bacterial cells as human cells. And then wait, the viruses are controlling the, yes, don't think about it too much. You'll drive yourself crazy, right? Okay, so as we jump into viruses here, right, infectious particles that are obligate intracellular parasites, one of the things that's going to become super clear is they are small, right? And so in this image, what you're seeing, this big blue sphere, is actually, there it is, a yeast cell, right? This is a eukaryotic cell. And for a size comparison, they've actually included a couple of the bacterial cells. And so seeing them under a microscope, they're tiny dots. And then everything else on here is trying to show you how big viruses are, right? So like this guy right here, this is a bacteriophage. You could fit thousands of them, couple thousands of them, right, inside of a bacterial cell. You could put 50 million, where's poliovirus? Poliovirus, right? Number 8 here. You could fit 50 million polioviruses in a single human cell. Okay, so these guys are extremely small. You don't have to memorize these sizes, but have some idea. Heck, here they show you a hemoglobin molecule, right? And we know how many millions of those are inside red blood cells. So viruses are small. Okay. So what I'd like to do is kind of run through the physical makeup of a virus, sort of like we did with both bacteria and eukarya. Now you should already be recognizing from this little chart that viruses are much less complicated than either bacteria or eukarya. And so that's kind of exciting. So let's look at this viral particle. It's really broken into two parts here, that outer covering and then the central core. So let's start with the outside there. So the outside of this viral particle is called the capsid. This capsid. It's made of protein. It's this protective shell that's going to be containing nucleic acids, right? So it's built from a bunch of protein subunits called capsomeres. And so each of these individual little dots in here that they're showing, oh boy, are capsomeres, and they come together to form this capsid. This is just trying to show you the main shapes that viruses will take on. This helical, it kind of looks like a rotini. Icosahedral, I say this one looks like the Epcot Center, right? All these different sides. And then the bacteriophages, and these, by the way, are the viruses. They only infect bacteria. They can't actually get inside of our cells. But so this bacteriophage, check out this. It's like an icosahedral on top of this little stem. And then what's with the creepy, like, spider legs, right? And think about how small this is. A couple thousand of these fit into a bacterial cell. It looks like a little lunar lander. Okay. So the capsid is going to be found on every virus, right? The protein coat, the protective covering. Now, some viruses have an envelope, okay? It's basically, notice, oh, see this right here? Between here and here, it's a phospholipid bilayer. Basically, the envelope is just a little piece of membrane stolen from the host cell. That could be cell membrane, nuclear membrane, endoplasmic reticulum, right? But some form of that membrane. It also has some of these proteins that are kind of embedded there. Oh boy. that are embedded into that membrane. They help kind of hold the envelope down to the capsid here. And the ones that are sticking out are referred to as spikes. These spikes we'll see become important in how the virus actually replicates. Typically we talk about H spikes and N spikes, right? So if you remember with influenza, you'll see things like H1N1 or H3N2. What they're talking about there are actually the specific spikes that are on the surface of this. So the H spike stands for hemagglutinin. I wish I could just write in here. It's in your book, hemagglutinin, and just think a glue. This is the spike that actually is going to help the virus dock onto the host cell, right? Stick to the host cell. The N spike, N stands for neuraminidase, and this spike is going to actually be important in how the virus, once it's done replicating, how it actually releases from the cell. So H1N1 has a different set of spikes than, say, H3N2. Little tangent, but while we're here, so these spikes can change, right? So you're going, oh, this is part of why there's like a different flu vaccine every year, because flu can change, influenza virus can actually change what spikes are on the surface, right? They kind of change over time. And so we see this happen really in two different ways. Where's my writer? So you have antigenic drift and antigenic shift. So an antigen is anything that causes the immune system to respond. This is terrible. Antigenic of the antigen. Antigenic drift is going to be small changes in what these spikes look like, right? So year over year, there's like minute changes that take place, but for most people, like their immune system still mostly recognizes it. They might have a minor illness, but their immune system should catch on and react pretty quickly. Antigenic shift is when you get a major change in those surface proteins, these spikes. So like H1N1, that was the swine flu pandemic, where no one's body really had seen this particular combination of spikes. And so a lot of people got sick and more sick than usual. So antigenic drift and antigenic shift. So those are the spikes. And okay, and so those are in the envelope. So that's it, right? The outside of the virus, they always have a capsid, and some viruses have an envelope. We call them enveloped viruses, which is very nice. The other ones are just naked viruses. Okay, the central core then of a virus is really just going to be where we have the nucleic acids. Now, I have to say nucleic acids because it can be either DNA or RNA, right? Now, you hopefully remember, right? DNA, it uses that deoxyribose sugar. RNA uses ribose sugar. And then you're starting to think about, oh yeah, I remember Gen Bio, we talked about all these differences between DNA and RNA. Well, one of the things that's strange here in viruses, right, is we are used to thinking of DNA as double-stranded. And absolutely some of our viruses, things like herpes, some of the pox viruses, chickenpox, for example, those carry in that central core double-stranded DNA. But there's actually some viruses that have single-stranded DNA. And so you're like, how does that even happen? It's using deoxyribose as the sugar, but there's just a single strand. So viruses can do that. Same thing goes with RNA, right? You're used to thinking of single-stranded RNA. Some of the retroviruses like HIV use that. But you can actually have double-stranded RNA in viruses. Things like rotavirus is a double-stranded RNA virus. Anyway, any possible combination you can think of, the viruses have figured out how to do this. Okay, the other thing that can be in the central core are what we call matrix proteins. So these are going to be enzymes that are already built. So this was built by the host cell, packaged into the virus, and sent with it. So these preformed enzymes, really that they need in order for replication to happen. So for example, polymerase enzymes. Do you remember the polymerases? They synthesize DNA or RNA. So you had like DNA polymerase 3 or RNA polymerase. And so some of those, in some viruses, those will come prepackaged. There's an enzyme, gonna love this, called reverse transcriptase. Right, the ACE ending tells you it's an enzyme. And you think reverse transcriptase. Okay, transcription was what? DNA to RNA. So then if we're reversing that, it's RNA to DNA. So this is an enzyme. This is, for example, is in HIV. This is an enzyme that allows it to actually take the RNA that it carries, right? It's a single-stranded RNA virus. And when it infects a human cell, it uses this enzyme, reverse transcriptase, to make DNA that human cells will pay attention to, right? And use to start building new copies of the virus. It's not things like enzymes for, say, metabolism. It is going to let the host cell produce all the energy that the virus needs, makes anything like that. But there are times where some viruses will carry certain enzymes that they need for replication. Okay, that's it. That's what's in a virus, right? They all have a capsid, some have an envelope, they all have nucleic acids, and then some carry some of these proteins. Okay, so pretty straightforward. All right, so what I want to go through then is how a virus actually goes about multiplying. And so this is true for animal viruses, and then we'll look at bacteriophages, which actually carry a lot of similarities. So you'll be like, oh yeah, that looks familiar. But this is specifically is for animal viruses. And so you might need to get your book out in order to read, right? So we've got a few steps here. We have adsorption, penetration. I might have fewer steps than you do. Synthesis, assembly, and release. Okay, so adsorption is when the virus actually attaches to the surface, right? And we were just talking about this. They use those H spikes to attach to the surface. So the surface of the host cell actually has to have the specific receptor in order for that viral protein, that viral spike, to attach. And so basically, these specific receptors identify the host cell. So with viruses, we talk about a host range. And what this looks like, so the host range is just the range of cells that can be infected. But this can go based on cell type or by species. So for example, if you think about rabies, right? What cell type does rabies infect? Anybody know that one? It's neurons, right? Brain cells. And then what species? It's not just humans, right? It's all mammals. So bats, humans, raccoons, old yeller, right? So that's the host range. And so basically we're saying that that virus can find the correct receptor in all of those species, but only on neurons. It doesn't do anything, say, to your skeletal muscle or your digestive tract. It can't dock to those cells. Okay, so then you look at something like COVID-19, right? We actually know the receptor, it's called the ACE2 receptor, and you happen to have lots of those in your lungs, and then also in your blood vessels. And so the virus is able to get inside those cells, right? Because they have the receptor. The virus is doing nothing, say, to your skeletal muscle cells, right? It doesn't dock there. Okay, so that is host range. And so you need the correct receptor to adsorb to. All right, from there, once the virus has actually docked on the surface, we're going to go through that second step. You'd have to go back. Oh, here we go. All right, if you want to see that. The next step then was penetration. And so there's a couple different ways that this can happen. So it varies from virus to virus. Basically, that virus is either going to use fusion or endocytosis to actually get inside of the cell. So fusion is what happens in enveloped viruses, right? Notice the extra layer here as opposed to this one. So this blue layer here, that is the envelope. And so really when they scoot up here, that envelope is a phospholipid bilayer. It more or less just like melts into the host cell membrane. And then we see the virus kind of popping out in the inside. So fusion only happens in enveloped viruses. Here we see this non-enveloped virus undergoing endocytosis. And this is where the virus is actually engulfed by the cell. So how mean is that? It like docks on the surface and manages to trick one of your cells into engulfing it. And then once it's inside, right, it's going to go ahead and spill open and release those nucleic acids and its enzymes. Okay, our third step, so they separated the uncoding, right, where the DNA actually gets out. I'm skipping that. So my third step is synthesis, okay? And basically what you have to have happen here is that viral nucleic acid takes over control of the cells, right? It is going to start using the cell's metabolism, the cell's machinery to build everything it needs, right? So it takes over transcription translation. So in this case, we actually see the virus is releasing this RNA. Heck, it doesn't even have to mess with the nucleus. All it needs to do is take over the cell's ribosomes, right? And the ribosomes will read that viral RNA and start making new capsimeres, right? Those are our proteins, new spikes, new enzymes, right? It'll also run DNA replication, not at the ribosome, but it can do, RNA replication. If you have the right polymerases, you make new copies of the RNA. So in synthesis, all we're saying is we're building all the parts of the new virus. Okay. Some viruses will actually, right, go into the nucleus. Some actually will insert their DNA into the host cell's DNA. And so that gets super strange. They can start altering the gene expression of the host cell. They can leave that DNA behind, and we'll talk about that. But there's synthesis, right, building. The fourth for me, fourth step is called assembly. And basically now all of those pieces, parts that we've built are just going to come together. So the capsimeres form that capsid, the nucleic acid is inside, the spikes actually move. Look at this, the spikes actually go out and embed themselves in the membrane. So see up here, it looks different. Here's the spikes embedded in the cell membrane. And again, how tricksy is this? Here we see our mature virus, right, at the end of assembly. It's then going to try to leave the cell, and that's our fifth and final step, release. And for this enveloped virus, it placed the spikes, it convinced the host cell to use the Golgi apparatus to put the spikes in the cell membrane. And now on its way out, it's just going to steal a little bit of that membrane. Spikes are already included. So that fifth step, that release, can look two different ways. So exocytosis or budding is what we were just looking at. And again, the spikes are already embedded in the membrane and it just takes it with them. Other viruses are going to use, budding is the same thing. I just thought this was such a cool picture. This is actually HIV leaving a cell that's just made new copies of it. And again, that cell might crank out thousands or tens of thousands of copies of that virus. Other viruses, instead of doing budding or exocytosis, they're going to cause lysis, just like what it sounds like. Sorry, this kind of came out horribly. All those newly assembled viruses are just going to rip out of the cell, splitting it open. That's much more common. That's the way non-enveloped viruses get out. And so that kind of makes sense, right? If you need that envelope, you're going to take part of the cell membrane, you're going to bud or do exocytosis. And then if you're a naked virus, you're just going to rip the cell open. Okay, so if we look again at that as a whole, think about this. Overall, this can vary in how long this viral multiplication takes, right? It depends on your virus. A few hours to a few days, right, for this viral multiplication to take place. But again, thousands, heck, I even wrote down up to 100,000 of those new viruses being released. And honestly, this is lethal to the host cell, right? Not only have you shut down its own metabolism, right? You're doing everything for the virus. You've potentially altered the host cell DNA, or you've destroyed the membrane, right, on your way out. So quite often, this process of viral multiplication is actually going to be lethal for the host cell. Sometimes it's not, and then all sorts of wacky things happen, right? So cytopathic effects. So pathic, you're thinking pathology or like illness. Cyto means cell. So what's going on with the cell here? This is referring to the virus-induced damage to the cell. And you can actually see this under a microscope. So this makes it really nice if you're trying to decide certain things about disease. You might actually be able to see it. So over here, they're showing you this example. They're calling this a giant cell. So basically what's happened here is these darker red sections are the nucleus. And so, where can I do this? Sorry, trying to see where my pointer is. So basically, these host cells have lost so much of their cell membrane that they're forming what's called a syncytia, where all the nuclei are hanging out together and hoping there's like enough membrane to kind of cover them all, right? This cell is not going to function well. Like this one probably should die soon. But that's something you might see. Often the cells will be like disoriented or misshapen or the wrong size, like this giant cell. You might see, so this line right here is the cell membrane of a cell. And all of those little spheres, we call them inclusion bodies. It's like masses of viruses or damaged organelles. And so that's going to be indicative of a problem as well. Other damage to the host cell. So you might actually have these visible cytopathic effects. Sometimes you get persistent infection. So what happens here is that virus actually finds a way to kind of hang out in what we call this latent state. It's like dormant, and it can remain inactive over a long period of time, right? It's then going to reemerge to some sort of stimulus. So the classic example of this is the herpes virus that causes cold sores, right? So you shared a water bottle in middle school, you got a cold sore, right? And then it might have been months until you get another one. And something like half the people never get another one. But basically that virus is able to lay latent in some of those neurons. And then, it comes back at convenient times, like when you're stressed or, you have a big event coming up, or you get a sunburn, or even it can be related to menstruation, etc. And so then the virus kind of reemerges and causes cell damage and does all its replication. The other thing that can happen is what's called transformation. And this is when the DNA of that host cell is actually permanently altered. And so what do we call a change in DNA? It's a mutation, right? And so this is going to be when that viral DNA is actually integrated into the host cell. And depending on where it ends up, that may or may not be a problem, right? But some transformations, if you alter, say, oncogenes or tumor suppressor genes, they can actually lead to cancer, right? So they might cause an increase in growth, divide indefinitely, right? Things that we might see in cancers. So there's actually some viruses that we refer to as oncoviruses. Onco is referring to cancer, and that just means this is a virus that can initiate tumors, right? HPV is the classic example there. It's been implicated in cervical cancer. Okay, we're getting there. So, bacteriophages. So these are the viruses that only infect bacteria. All bacteria that we know of are parasitized by some virus. And what gets weirder is that bacteriophage can often make the bacteria more pathogenic, right? More infectious or more harmful to humans. And it just comes back to they're moving DNA around, right? And so sometimes that happens to help this bacteriophage. It's not like premeditated or anything like that. Okay, so let's look at this lytic. Oh, I was trying to circle, not cross it out. Let's look at this lytic cycle here. Let's go ahead and clean that up. There you go. Okay, that's where we are. That lytic phase. Take two. Lytic. Boom. All right. So this looks almost identical to what we saw happen in animal cells, right? So there's adsorption. It has to dock on the host cell. We have penetration. It has to get in there somehow. But what we'll see in these bacteriophages, instead of like getting engulfed, our freaking little spaceship lunar lander thing sticks out a needle, pierces the cell, and injects its nucleic acid. This is freaky. Okay, so that's penetration. It's actually injecting its DNA. But then the bacterial host cell goes ahead and replicates all the viral components. Those components assemble, and then it's going to leave the host cell through lysis. Okay, so that actually looks the same. They're just never enveloped, so they always are going to use penetration to get in, and they always use lysis to get out. Okay, this is what gets a little bit weird when we get over to what is called the lysogenic state. And so they're basically saying, okay, there's two different states of being for our non-living viruses, right? One, it's infecting these host cells like this lytic phase, making new copies, infecting another host cell, making new copies. The lysogenic stage, what happens here is you get the DNA being injected, and then we can enter this lysogenic stage. The DNA, the viral DNA is in blue over here. The viral DNA is actually inserted into the host chromosome. Okay. They will now refer to this as a prophage, right? So it's like before being a phage. So we have this latent prophage. Now the bacteria holds on to this genetic information, and every time it replicates, it passes it on to its offspring. Bacteria replicate really rapidly. Like you can go from 1 to millions in 24 hours. And every time they make that copy, they copy the viral DNA as well. Again, super tricksy here. So in this lysogenic state, we'll get to a point where they call it induction, where that bacteria, or sorry, I guess the virus moves back to the lytic cycle. So think about it. This bacteria just went through, hundreds of copying events. We now have millions of bacteria with the viral information, and then boom, it's like it pops right back in here. And now the bacteria cell starts making viral components, and we're going to have that many more popping out there. So this is pretty wild. This is something that only bacteriophages do. They'll also, there's an actual picture of the bacteriophages all over the surface and coming out. It's really creepy to me. The other thing to keep in mind here, right, is that this bacteria sometimes undergoes transduction, which is very similar to the transformation we were talking about with animal cells, but we call it transduction. And this is when the phage transports bacterial genes from one host to another, right? So when this bacteria starts producing viruses, if a little bit of the bacterial genes come with it, then when it infects the next bacteria, it can have, right? And so transduction is actually really important in the evolution of bacteria. We'll see things like antibiotic resistance getting passed this way, or like new toxins being produced by certain bacteria. Okay, so viral infections, right? There's billions of viral infections every year. They can cause death, illness, chronic afflictions, right? We looked at some of the oncoviruses causing things like cancer. We won't be working with viruses in the lab because they are obligate intracellular parasites. They always need to be living in a host cell. And so very commonly, they'll actually use chicken eggs, right? That's a cell to replicate viruses. It's way more complicated than what we want to do in lab. I guess I should say this. So when you're trying to detect viruses, right, there's a few different ways. A lot of people have been talking about these antigen tests, right, trying to find out if you've actually had COVID. You can use a technique called PCR, where you can take really small samples with just a tiny bit of viral material and see and detect infection that way. You can use, like we were just looking at the histology, some of those cytopathic effects can be indicative of certain viruses. Your production of antibodies, there's the other big COVID test, right? If you're producing the antibodies, we know you've had it, right? Typically that's how HIV, for example, is detected, is actually through the antibodies. As far as treatment, there are antiviral drugs. So we've actually learned a lot about these through HIV. And so you've got certain drugs that can block the ability of cells, say, to make new nucleic acids for the virus. So basically you have to target the host cell in some way to stop that replication. Okay, so the last topic in this chapter is really prions and viroids, and I would be remiss not to talk about them because they are fascinating. So prions, you guys are probably familiar. Mad cow disease is the classic example of a prion disease. A prion is a infectious protein molecule, right? So prion is like infectious and protein kind of stuck together. Okay, so all of these diseases fit into a category called transmissible spongiform encephalopathies. It's in your book. Transmissible spongiform encephalopathy is fortunately abbreviated TSE, which is easier, right? But there's all sorts of diseases that fit in here. So scrappy, you'll find in sheep. Mad cow disease, the technical term is bovine spongiform encephalopathy, or BSE. If you're a hunter, you've probably heard about chronic wasting disease, CWD, in elk and deer. There are human variants of this. So there's one called Creutzfeldt-Jakob disease, CJD, Creutzfeldt-Jakob disease. And this is actually the human version of mad cow disease. And so, for example, in Great Britain, there was a big outbreak of mad cow disease, and you actually can't donate blood if you've lived in Great Britain in that set of years, because they're worried that you might be infected with CJD. There have been somewhere around 100 deaths in England, but some of these prion diseases can take years to turn on. And so, yeah, there's continued concern there. There's a TSE called Kuru that is in a certain tribe. It's the Foray tribe in Papua New Guinea, and they practiced cannibalism. And so part of the death ritual, it sounds like, was actually consuming the brains of the deceased, right, as a way to like pass down something. I won't pretend to know the details of the cannibalism there, but basically by eating that infected nervous tissue, it was passed on, right, to the next generation, but it doesn't show up right away, because what you have to do, what happens in all of these prion diseases, is notice, like, this is normal brain, and then a prion has all these holes in it, like these big openings. That's why it's spongiform. So it's transmissible. It can be caught. Spongiform. Encephalopathy. Pathology is illness. Encephal refers to the brain, cephal, and then it turns your brain into a sponge. But sometimes this takes years. There's an inherited version called fatal familial insomnia, or FFI, and that attacks the portion of the brain that lets you sleep. And so these people, when the prion starts causing all these holes, they stop being able to sleep, and they actually die from insomnia, right? And so when you're thinking about prions, if this is what the protein is supposed to look like, it's really just a misfolded protein. And so this is weird, right? Like, viruses were strange enough. It was like a protein coat with some nucleic acid in the middle. Now we're talking about just a protein that can make you sick and kill you. And so it seems like when this abnormally shaped protein comes in contact with another protein, it can cause it to change shape as well. And so if you ate, say, the brain of an elk with chronic wasting disease, if that misshapen protein got into your nervous system, it could start teaching your proteins to misshape as well. I don't know that there's actually been a confirmed human case of picking that up from deer or elk. Okay, so the other thing I'll say about prions, right, is it's a protein. You can't kill a protein, right? It's not a living thing. And it's already misshapen. So these don't, like, normally we put things in the autoclave, right? At high temperatures, proteins denature, they no longer function. That doesn't seem to work against these prions. So there actually have been incidences where medical equipment, right, surgical equipment that has gone through the sterilization process and used on subsequent surgeries has actually passed some of these spongiform encephalopathies. So they're a challenge. This all sounds very scary. It's also very rare, right? This is not the biggest of our problems right now, right? Okay. On a happier note, viroids, they only infect plants. So a viroid is just naked RNA. No protein coat. naked RNA, and it only infects plants. And so they're just trying to show you, hey, happy plant, viroid infected plant, happy plant, viroid infected plant. And so these are important agriculturally, but then there's also not really anything we can do about them. So, but they only infect plants. So that's your happy note to end on. Okay, I have talked way too long, but I hope that really kind of helps you with chapter 5. Viruses are the big picture, and then a little bit about prions at the end there. Okay, I look forward to seeing you on Thursday. We can talk more about this. Definitely write down if you have any questions or things you want to go over more. All right, bye.